everyone, welcome to STEM News Nexus at UCLA, blog of preprints and new publications. I hope everyone had a great week 2 and are looking forward to week 3. So let's get started on my paper for augmented allosteric maps by this paper in Nature entitled Mapping the Energetic and Allosteric Landscapes of Protein Binding Domains, published on April 7th of 2022, and these are the pages I'm referencing. So essentially, here's the background. There are many regions of the protein around the active site that can react with different chemicals to change the role of the gene product. So this could be interacting with different proteins, and also just general chemicals that are not amino acids in the cell. Medications that target these sites can be effective can be more effective than targeting active sites themselves, but you can basically look at this protein structure with a bunch of different switches, and based on the different proteins that bind to it or different chemicals, that will flip the different switch so that that gene product has a different function. But if we look at just specifically those like protein binding switches, they also have switches around them that kind of affect how that protein interaction is going to change the gene product's function, and with the state of the cell in consideration. So we don't have maps of these allosteric sites for that affect the protein interaction, and tracking mutations of these sites can be helpful to ascertain the function of these sites around the protein binding domain, or the protein dependent switch, if you will, excuse me. And so this can be helpful, but it's complex because the specific mutations can affect the biophysics of the protein in different ways. Like it could actually make the proteins have a greater affinity to the site, and that can change the stability of the protein, or just a greater amount of the protein can bind to the allosteric site per time, which can also have a different biophysical effect that could then manifest into a different role of the gene product, or basically flip the switch in different ways. So the different mutations in the allosteric sites can flip the switches in different ways to do different functions. So they are asking if they can track these mutations in a non-ambiguous way that allows them to map the allosteric sites for protein interaction sites. So they decide to sum up all the binding and affinity reactions associated with different genetic variants of allosteric sites around a protein binding domain, and fit them to thermodynamic models using neural networks. And they decided to test this method on these well-known SH3 and PDZ binding domains. For example, from this passage from Molecular Biology of the Cell, so we have these claudulin proteins that have membranes in tight cell-to-cell junctions that prevent fluid go through. They actually have different mechanistic roles. So here we have the claudulin, but all of these different proteins actually have different PDZ and SH3 domains that can interact with different proteins within the cell, depending on what the cell is doing or what kind of cell it is. And that can then have different responses. So on these like particular domains, depending on everything around it and everything that's interacting with it. So from applying this method on these particular domains, they saw that mutations could affect how specific interactions involve with each other instead of removing interactions. So if they have this allosteric site around a switch, it's just going to affect how the proteins are interacting with the allosteric site, or the chemicals are interacting with the allosteric site, as opposed to just removing that interaction altogether. And they also see that more of the mutations are effective or have an influential role in the protein binding domain if they are closer to it. And then they have different glycine residues, and then that are also specific towards affecting the protein-protein binding domain. And also they have specific residues within the PDZ binding domain that seem to have different effects. So they conclude that they can use this method to map the allostericity of many different kinds of proteins. So that's just something that we'd have to continue to look with their literature. So if you want to read more about it and look at their models, feel free to look at this publication, and make sure to sign up for our newsletter for more updates on STEM news. Anyways, thank you. Have a great week.